started it?
Yeah.
It's recorded, okay. So I guess I'll get started then, please. Hi everyone, it's a great pleasure to introduce Olga Vitek this afternoon. And I've known Olga for many years, and she has really brought a lot of statistical rigor to the field of proteomics. So she started out with a PhD in statistics from Purdue University, and then she did a postdoc with Rudy Abersol at Institute of Systems Biology. With this strong statistics, she approached Rudy and said that she can do the statistics better than he can. He listened and said, "Show me," and she succeeded doing that. So then she went back after her postdoc and took a faculty position first at Purdue, where she got tenure and developed many different statistical methods that were applied in mass spectrometry based proteomics. And then she recently, a year and a half ago, joined Northeastern University and has moved her lab there. So welcome.
Thank you so very much for the kind introduction. It's really a pleasure to be here, and it was really nice meeting everybody in the morning. So as David mentioned, I'm a statistician by training, and we are interested in all things quantitative mass spectrometry and proteomics. And we have a variety of projects in the lab. But today I decided to talk about one particular project which focuses on profiling, on quantitative profiling of biological samples using mass spectrometry experiments. And so last night David suggested that some of you may actually need a brief overview of quantitative workflows and mass spectrometry. Do you mind having a show of hands? How many are very, very comfortable and would be annoyed if I talk about what mass spectrometry is? Okay, let me phrase it the other way. How many of you would like to have a couple of words about quantitative workflows? How many of you don't care? Okay, well, because of that, so I will try to go fast, but please don't hesitate to interrupt me, okay? And from the question, I will figure out if I'm annoying you or not. So, but I have a point here, though. So I would like to tell you a little bit about that to motivate the reason why statistical methods are needed and why they require a lot of complexity. So for those of you who need a lot of refresh, and I don't know how many of you are here, just to kind of set the stage, right? So we are interested in, oh, there's no mouse. Okay. We are interested in system-wide characterization of proteins, ideally all of the proteins in the system. And by characterization, I mean sequence structure, localization, abundance, PTM, and so on. And it is more challenging than gene expression on one hand because of the complexity of the problem, because, for example, if human genome has 20,000 protein coding genes, if we look at the translation splicing proteolysis, we have a much larger number of proteins. And then if we add DNA rearrangements and PTMs, it's an even larger number of protein instances that we may be interested in. And of course, another problem is the dynamic range, that in plasma we have over 10 orders of magnitude. And unlike messenger RNA and nucleotides, we cannot make more of the protein. We need to study proteins which are present often at low concentrations. And so the way to make progress is twofold. So one is obviously on the experimental workflow side in terms of sample preparation, separation, instrumentation, and so on. But I would also, of course, would like to argue that statistical methods of design of the experiments and analysis of data can really contribute a long way towards objective interpretation of the data and also sensitivity and reproducibility. And so very briefly, in terms of numbers of proteins which we can study these days, so this is taken from a review which is somewhat old, but I think it gives us an idea that on one hand, as time progresses, there is more and more publications involving proteomics. In the last decade, increasingly the progress is made by characterizing the percentage of proteome coverage of different organisms using proteome, and in terms of the number of proteins which are reported by different studies. So you see that increasingly the numbers go very high into hundreds and thousands and even tens of thousands of proteins. However, the majority of experiments will stay between 200 and 1,000 proteins. So this is kind of the space that we operate with. And just very briefly, for technological purposes, we don't study proteins, we study peptides. And we ionize peptides by attaching charged molecules essentially to them. And then we place the charged peptide molecules, for example, into electromagnetic field. And then peptides which are heavier, they travel longer. Peptides which are lighter, they travel faster to the detector. And we can use this to essentially back calculate the ratio of mass divided by charge of the peptides, and the height of the peak corresponds to the amount of the biological material which was detected. And the modern workflows go beyond that, because of course in complex mixtures we can have many peptides which have the same ratio of mass divided by charge. So what we also do, we add a separation step where, for example, we separate the peptides by property which has nothing to do with mass or with charge. So for example, hydrophobicity. And then if we place the peptides in the environment where we essentially vary the properties of this environment, then peptides which don't like to be in water leave earlier, peptides which like to be in water, they leave later. And then at different time, we have different species of the peptides which are separated by mass divided by charge. And now we can reasonably expect that individual peaks correspond to a single peptide species. And this does not yet give us the information on the identity of the peptides. For this, we need an additional step where the mass spectrometer selects peaks, and traditionally this would be peaks which have highest intensity and subject them to high energy, and these peaks, the peptides fragment. Like for example, here is a cartoon where we have a peptide sequence. So if we subject peptides to high energy, then it breaks into small pieces. Each of the pieces has a mass, many of them have a charge, and so now this creates a secondary MSMS or MS2 spectrum. And so this is used for identification of peptide sequences from the same experiment. So this is like in very, very, very brief summary what we are working with in a very basic form. And so this particular workflow is called shotgun or data-dependent acquisition. And it is data-dependent because we usually select the highest peaks for fragmentation. And so depending on the height of the peak, some of the peaks are selected for fragmentation or not. And so the actual raw data would look like that. So now if this is the ratio of mass divided by charge, this is the retention time, so the time at which different peptide species went into the mass spectrometer. So the peaks which come out of here, the height of the peak is related to the abundance of the peptide, and some of them are selected for fragmentation and have SMS spectra, which are informative of the identity of the peptides. And then for computational computational steps start here, and so we have two types of workflow. On one hand, we have MSMS spectra, and we have the database of theoretical peptide sequences which can be predicted from the the way the peptides are obtained, so from the digestion. And it is possible for the organisms for which the genome is sequenced. And so we can map the peptides in the database to the observed spectra to identify peptide sequences, and then also map them to the parent protein sequences. On the other hand, we have this part of the workflow where we need to locate the peaks, map the peaks to the identified peptides, and then map the proteins and align the peaks between the runs. And from this point on, we would have structures of the data which are familiar to many statisticians, where we have, for different features, we have measure of abundance across different replicates. And we would apply some normalization, transformation, summarization, and then the actual analysis starts from here. So this is really a very, very brief summary of the type of methods, the type of experimental workflows that we use with our data. But of course, this is just the peak of the iceberg, because as I mentioned earlier, the challenge is that many analytes are very low abundant. And when we work with the type of workflow that I just described, we're very limited in terms of how many peptides we see and what are the abundances of the peptides which can be visible. And so the progress is made by, on one hand, changing what we place in the mass spectrometer, so what type of samples we have, and on the other hand, how the experimental workflow proceeds itself. And so this is just a few examples here. So what I described right here would be this type of experiment. So let's say this, we have one healthy and one disease subject. So we have proteins digested into peptides. And so then we can have one peak for the peptide in the healthy sample and a corresponding peak for the peptide in the disease sample. And if this peak is selected for fragmentation, we would also have the corresponding MSMS spectrum. And so quantitatively, we can do two things. We can either compare the peak intensities between the two runs, or we can even use the number of MSMS spectra which were produced from each of the samples. And because the instrument selects the peaks for fragmentation as function of their abundance, the count of MSMS spectra can actually be thought of as a proxy of the abundance. So at least two ways to quantify things: count MSMS spectra or integrate the peaks and compare peak abundance. Now we can do other things. So for example, if we want to quantify multiple samples simultaneously, and this is what is called multiplexing, we can do things such as label the components of the samples in different ways. For example, this is metabolic labeling, meaning that we have a label attached to the peptides in one of the samples. And a label in our context means that we modify the mass of the peptide. And so now, if we acquire this mixture of two samples together, instead of one peak that we had before, we have two peaks. And they have the same time because their hydrophobic properties did not change, but they have a known difference in mass. And this difference corresponds to the mass of the label. And so now we have two peaks in the same space, and we may want to compare the two peaks together. And this is advantageous because now we can quantify two samples in one run. It has also advantages from the statistical kind of perspective, because if there's a lot of run-to-run variation, this variation simultaneously affects the two peaks, and then we can kind of eliminate this impact of run-to-run variation on relative quantification. So another approach to do this is just to add reference standards, and this is particularly useful when we have a lot of technical variation. In this case, unlike here, where we're labeling two biological samples of interest, we're labeling a standard peptides which do not have any biology in them, and we just add them to the mixture for the purposes of normalization between run. And so now we also have two peaks, but unlike here, this is the peak of interest, and this is a technical kind of standard which we use to reduce the unwanted variation. So now we would have this pair for the disease sample and a similar pair for the healthy sample. So this is what can be done. But then there are additional things, and there is an isobaric peptide labeling, iTrack, and more recently TMT, where we can have multiple samples labeled, but now they're all mixed and they all give rise to a single MS1 peak, but then the difference occurs not at this level, but at the level of MSMS spectra. And so now we use the reporter ions in the MSMS spectra to quantify each of the samples separately. And again, we know which peak came from which sample because they are labeled with different molecules which have different masses, and so we can use this mass shift to infer which peak came from which sample. So the reason why I cannot talk about all of that is that as a statistician, I'm really interested in the structure of the data. And those of you who have some statistics or computational background, you'll probably see right away that these are very different types of stochastic variation. And so if we want to make conclusions, and for the purposes of today, I will just talk about what is changing between healthy and disease, real simple, or any two conditions that we have. So for example, these are count data, right? And these are continuous data. So usually this has to be modeled in a different way. And if you're familiar with transcriptomics, so we know there are also settings where, for example, microarrays give us continuous data, next generation sequencing, RNA-seq, gives us count data, And so we also have to change our models that we use to analyze this data because the structure of the data is different. Unlike in genomics, spectral counting is less accurate. So in RNA-seq, count data are more accurate than microarrays. So in our case, peak intensities are more sensitive and kind of have better quantitative accuracy than spectral count. So I will focus on continuous data for today. So this is what happens when we start preparing samples in different ways. And so you can see right away that we can multiplex in different way. We can have many samples mixed together, or we can purchase standards. So clearly, these are all different costs, different times, different accuracy, and different data structures for statistical analysis. But this is, again, not the whole story, because another thing which is very important is how this is quantified. So we see that here just counts of spectra, MS1 peaks. Here we have some reporter ions in MS2 peaks. But now there are more recent types of workflows which actually proceed by fragmenting MS1 peaks into pieces of peptides, but the quantification occurs at the MS2 level as opposed to MS1 level. And how it happens, well, depends on the specific workflow that we have. So here I have SRM, it's called selected reaction monitoring, where within the instrument, the fragments of this peptide are isolated separately, and you can quantify the abundance of the fragments. And this allows us to gain sensitivity. It allows us to work with lower abundant molecules. But there are other technologies, more recent, which is called data independent acquisition, or DIA, where the individual fragments are not isolated within instrument, they're all fragmented jointly, and then you have the same type of quantification conceptually, but there are additional computational issues involved in determining individual peaks. As a statistician, what I see now that, again, it changed the structure of the data, because now we have the fragments which are nested within the individual peaks. And so now we'd also need to make sure that we account for this nested nature of the data structure for quantification. And so just to kind of give you a summary, and for those of you who come more from the computational quantitative background, you will be very used to that, so that at the end of the day, if we have count data, we will just have a particular protein, and we will have multiple sample times and one kind of count of MS spectra per sample. On the other hand, if we work with peak intensities, we will have multiple samples and multiple groups, and now we'll have multiple features. And the features will correspond, on one hand, to multiple peptides. Remember, we have a protein which we digested into peptides, so one protein can have multiple peptides. And then within one peptide, if we do targeted experiments, we can have multiple fragments. So all of these features will be part of this data structure. And so now we can potentially have a complex experimental design. And this is really simple disease and control, but modern experiments are much more complicated than that. And so you really want to compare things in a more complex way, for example, time course or paired samples or some complicated combinations of conditions. You also want to integrate the measurements across all the features by taking into account the complexity of the data structure. And at the end of the day, ask questions such as, is there a change for this protein between disease and control, or group 1 or group 2? And so this is the type of question that I would like to talk about. And this is one part of the work in my group. And so MSstats is the software which we started a long time ago when I was still a postdoc and I became a faculty. And over the years, this became a fairly complicated or comprehensive project which combines multiple functionalities. But the scope of the work is, our goal is to provide a tool which focuses on statistical relative quantification of proteins and peptides, which essentially asks the question, which proteins change in abundance? Our goal now is to make it as general and as broad as possible. So we try to develop methods which work with arbitrary complex experimental designs, because different research questions requires different design. And so one person may have 2 groups, another has 5 groups, and another has some complicated factors. We want our users to be able to analyze their data in all generality without necessarily knowing the details of how to model the data, and also how to kind of have very sophisticated use of programming languages. And by the way, I should say right away that everything we do is open source, and it is in R. So we use R programming behind that. So we aim at addressing really general situations with complex designs, factoring experiments, and so on. Now, we also aim to provide tools for people who use diverse workflows. And so for the moment, it is limited to chromatography-based quantification. So essentially, we do not kind of work with iTrack or TMT. But everything else, such as shotgun data-dependent acquisition, targeted SRM, data-independent acquisition, DIA SWATH, it can all be handled in all generality within this approach. It can be label-free or label-based. So for example, if you use this labeled standards, you can still analyze this data with MSstats. And the functionalities that we offer would be data visualization, statistical modeling, inference, sample size calculations, and things like that. And we work very hard to make it accessible to practitioners by making it interoperable with other tools that people use. And we work very closely with Skyline to integrate. So if you're familiar, Skyline is a computational environment, especially for targeted and data-independent acquisition experiments. And we also have converters to other tools such as MaxQuant, so that you can use the MaxQuant output and work together with MSstats. So let me tell you, and so the point here I would like to make with the next part is twofold. So if you come more from the experimental background, my hope is that what we do is useful for you. If you come from a statistics or computation background, my hope is to show that you can get very far with very simple things. And often simple is good, because simple means more general, and the hope is that your users do things in the right way more frequently, right? Because oftentimes complicated methods, as somebody said recently, complicated methods fail for complicated reasons. And so my hope is to say that, to show you that even if maybe you're just a beginning student or just started working in this area, you can go a very long way even with basic tools. But then of course there are also research challenges there. So this is one example of a label-free experiment, and this is an example of a complex experimental design where we had two cell lines, low and high invasive. The cell lines had low oxygen or normal oxygen, and the oxygen treatment was kind of administered for 2 different time points, and then there were different cell cultures of the cell lines. So all in all, it's a factorial experiment with this many combinations of conditions. And this is one particular protein from this data set. Different colors correspond to different peptide ions here. Different points are the replicate runs and replicate cell cultures, and the lines link the means in each condition. And so the question is, I want to know what is changing between different conditions. And very clearly, you can see the difference between the two groups here, which is low invasive and high invasive groups. And fine, right? But maybe you want to know things such as, does oxygen exposure matter? Does the high invasive cell line respond differently to oxygen treatment than the low invasive cell line? So you may ask questions which are more complicated than just low versus high invasive. And so when we started this long time ago, our collaborators, they would literally take 2 groups. They will work with 2 groups separately, and they would do something like t-test, and then they will start over with another 2 groups. And my message is that this is ineffective, and you can do a much better job by modeling all the data simultaneously and combining the information from different conditions to still answer the question that you had in mind. And so this is how MSstats works. So we start from the experimental design, and really we should not underestimate this step. This is really important, because if the experiment is not designed correctly, nothing else matters, really. No amount of statistical sophistication will save problems with bias or some errors and so on. And I will come back to this later, but for the moment, at least, we need to understand what kind of experiment this is. Do we have time course? Do we have multiple groups, paired design, and so on? So here's another example of another collaboration we had. So this was samples from childhood cancer, where we had some healthy controls just at one time point, but then the disease subjects, they actually had measurements of plasma before treatment, during chemotherapy, after surgery, and so on. So you have a combination of 2 groups, but one of the group has a time course. So you can actually analyze this type of data with this tool. The next thing, and again, this is the second most important thing, is quality control and normalization. And again, I cannot stress enough the importance of that, because this is really what would impact greatly the results. And this would be an example if you only have endogenous peaks, so no standards or no labeling. And it's really hard to know if this change between these are 10 time points, if the change in time was due to some kind of artifact of the experiment, or was it some interesting biology. If you are familiar with genomics and how people normalize genomic data, so usually the assumption is that the majority of the analytes do not change in abundance between conditions, and people equalize medians, or they would do quantile normalization, or things like that. This really makes two assumptions that, one, the majority of things does not change, and two, the number of analytes is very large. In proteomics, more often the number of analytes is smaller, and depending on the experiment, especially if it's a targeted experiment, a lot of things actually change, in which case the traditional normalization methods do not apply, and you really need to rely on standards for normalization. And in this case, it turns out this is the actual biological response, and if you apply constant normalization, you will essentially normalize out the actual biology from that. And so a way to do it, so this would be an example with reference standards. So now you know that the standards do not have any biology. And so any shift that you have is due to the technology. And so equalizing the medians in the standards and applying the same shift to the endogenous peaks, this will be a much better approach. Again, the problem is that this will only normalize away the artifacts which occurred after you added the standards. And all the problems upstream will not be accounted for. So normalization is a really, really challenging aspect here. So as far as the tool goes and the implementation, we essentially allow the user to tell us what the standards are, and if they have a separate standard for each endogenous peptide, or if they just have a single or several common standards for the whole experiment, and we can use that as normalizers prior to the analysis. So this is the second part, and then the third part is statistical modeling, and this is really where statisticians contribute a lot, and this is where you can really leverage all the statistical techniques for synthesizing the information across different features, different replicates, different peptides, and come up with hypotheses that you can test, which would be various comparisons between different groups. And so the first thing to do is to describe the statistical properties of the design. So what the design is, what are the sources of biological variation, and what are the sources of technical variation. And of course, verifying the assumptions is a very important part. So this is what we decided to do. So probably if you look at this representation of the data that we looked at before, it's an array, right? And this is something which is very familiar to statisticians. So we have multiple conditions. Then we have, so this is a special case of group comparison. So let's say we have no time course or anything like that. We have distinct subjects in a healthy group and some other distinct subjects in a disease group. And so now in statistical language, subjects are nested within conditions because these people are different from these people. And then if we do acquire technical replicates, it means that runs are nested within subjects. So all of these runs belong to subject 1, all of these runs belong to subject 2, and so on. And by the way, I forgot to mention, but I do hope that you realize that it's really important to randomize the order of the runs. So I'm putting it here for illustration, but this is not the order of acquisition, right? This is just the order of we'll look at. So the randomization is really, really important for the experiments we talk about. For any experiment, but for proteomics, it can really make a big difference. Okay, and then we have the features. And so this is a very kind of traditional thing, and probably if you have had any statistical methods in the past, you immediately think things like analysis of variance, right? Or how can we summarize this information? And this is very appropriate, except that if we also look at the subjects and the runs, so we have multiple layers of variation, where we have the biological variation and technical variation, but these linear models are very easily extended to situations with multiple types of variation. And this is something which we proposed some number of years ago, where we developed a model which summarizes all the sources of variation from this type of array. So there is some kind of baseline. This is the contribution of the condition. So it expresses the fact that in some condition the protein is more abundant than other. This is what we're interested in. And then subject nested within condition. And then we have run-to-run variation. And of course, different features have different intensity because they ionize differently, right? And then we have the difference between run and feature variation, which is essentially a random error. And we have different constraints, which are necessary for linear models, and so on. And by the way, I forgot to mention that these numbers here, this will be log intensity of the peak. So we are working on the log scale. So by looking at the difference between conditions is equivalent to look at the log ratio of protein abundances between conditions. Okay, so this is what we did. And we actually, the older versions of MSstats went through quite a few versions already. And our old versions pretty much dealt with that. We recognized two things. As the technology improves, the feature-to-feature variation becomes much smaller than run-to-run variation and the biological variation, because the instrumentation improves, the signal processing tools improve. And so the reason why it becomes smaller and smaller is because, again, if you're familiar with analysis of variance, the full randomization would be you acquire this feature in this run, then this feature in this run, and then this feature in this run. This is not how the experiment works. You take one run, you acquire all the features together. Next run, you acquire all the features, and so on. So essentially, all the features are acquired together within a run. And now with modern tools, both instrumentation and signal processing, the feature-to-feature variation within run is much less than the run-to-run variation. And this is important to be recognized. And so now we need to account for the fact that this feature variation does not represent the full extent of technical variation, but only a subset of it. So this was the important aspect of experimental design. And kind of on the technical side, when you have a model with this many variance components, technical error, run error, subject error, we also had convergence problems. And just estimating this on a large scale from many proteins over and over was challenging. And so recognizing these two things, we understood that this is really should be viewed as an instance of a split plot design, which is also a fairly traditional design in statistical literature, where we have one source of variation within run in this case, and then we have another kind of type of variation. So essentially, this variation is due to technology, and this variation is due to biology. And run is the link between them. And we really need to recognize that, and recognizing that means it changes the inference procedure, and it also allowed us to change the estimation procedure. And so what we do now, we start from the subplot, where we ignore the source of each run. We ignore which sample originated each run. And so we just look at this as a two-way analysis of variance type of problem, where we have features and runs. So this is purely technology. There is no biology whatsoever. And then the inference from the split plot will be based on the summary of feature intensities within the run. The summary can be obtained in many ways. Traditional methods will just take the sum of the intensities on the original scale without taking any log. Maybe you can take a log and take the mean, or there are many other ways. So our approach here is to recognize that there are outliers and things like that in this data. So we use Tukey median polish, which is robust two-way ANOVA, and we use Tukey median polish to obtain a summary of feature abundances within each run. And so this is the intermediate data structure which we obtain here. And after that, we concentrate on the biology. So now we have one number per protein per run. And here we can do the work expressing the actual experimental design, dependencies between samples, and so on, to compare the conditions. So this two-step estimation had two advantages. One is it recognized the difference in type of variation. And two, it broke the problem into two steps, which now allows us to do a lot more work on the summarization of intensities within one run than we previously thought we could do. And so this really made a big change in how we summarize the abundances of protein in the run. So to kind of repeat it once again, so the current approach is to do the summarization of all the feature intensities so that we have one number per protein per run. And then we use this number as input to a model which focuses specifically on the biology or on the clinical aspects. And so this is the current approach. So just a little bit more on Tukey median polish if you're not familiar with that. So this is how it works. So if this is feature and run, so subtract column median from each value, subtract row median from each value, iterate until no change. So then what is left in this table after subtracting all of that is the residual of this robust fit. And then fitted values would be take the original values and subtract the residuals. And then the summary would be average over the column will be the run. And so this is the robust version of two-way analysis of variance, which is less sensitive to outliers. And that's kind of what it looks like. The problem is that this approach does not account for missing values well enough. And this is what our benchmark results showed. So to add additional kind of robustness to missing values, we have another step now. So if some of those intensities are missing, what we do, we use approaches which are used in survival data analysis, where you assume that some data are censored because they are below a certain threshold, and we use regular additive model to impute the missing values based on the censoring approach, and then we reanalyze this with Tukey median polish to eliminate sensitivity to outliers. So it's kind of two-step iterative procedure. And again, there's really no deep theory for that besides the fact that our benchmarking efforts show that this is the most accurate way to go. So that's what currently we're doing in MSstats. And this is just to illustrate some of the results. So this is a data set which was acquired 2 years ago as part of the Association for Biomolecular Resource Facilities, ABRF. There is a study group, IPRG. And the study from 2 years ago was exactly that. So can we have a controlled mixture where we place proteins in complex backgrounds in different concentrations, and we can see if we can detect changes in abundance, and we know the answer, right? So we can compare different methods by their ability to detect the known answers. And so this will be just some examples. And so we do several things here. So in gray, these are the original features, and in color, and you don't see it much because this is an example of a protein where everything is perfect. It's actually a background protein, so nothing changes, but there are also no problems, no outliers, no missing values. And so the green is the most interesting to us, is log of the sum of the intensities, where the sum is taken on the original scale and not on the log scale. Linear model was the previous version, which just was based on ANOVA and mixed models. And then TMP is Tukey median polish without censored-based imputation. And this is what we propose. And so essentially here the point is that, so true-false changes 1, nothing changes. When there's no problem, there's no problem. Everybody does very well, right? So
where is the linear model?
It's actually equivalent to the other one, so it's kind of hidden there.
Okay, and TMP as well.
Yeah, so they're all over. So the only thing you really see is the green line, because the green line is the sum. And so it will always be above the actual features because it adds up values instead of the mean. So essentially here, just to kind of show you the readout, right, when there is no problem. There is no problem.
That's one of the benchmarks.
Sorry?
That's one of the benchmarks that you use.
So this is one of the benchmarks. So this is a data set which has a large number of background proteins, and I believe 5 or 6 spiked proteins at different concentration. And so this is the background protein, which true-false change is 1. And so it's fine, right? And everybody, well, so there's a little bit of dynamic range kind of issue, right? It's not exactly 1, but everybody does the same thing, right? Okay, and it's not differentially abundant, so everything is good. So now this would be an example where we have outliers in low intensities. And this is very common, right? And so here is an example where it's a missing value, which if you impute this with 0, so that's what happens, right? Or this is not an imputation, this is just essentially a peak which was maybe misidentified or misquantified somehow. So now you start seeing gray lines, so these are the original features. This is the sum, which looks like that, and it's above everything because you add things, right? And you now start seeing that you have the, so the proposed in TMP, they are identical if you have no missing values, right? So the interesting thing is linear model. So linear model will be definitely affected by this guy, right? But it's also affected by some other outliers here. And you see already, so the true-false change is 1. So comparing condition 3 to condition 1, the true-false change is 1. And so now, in this case, TMP improves upon linear model. So here, if you use the imputation, there is no missing values, and so it's the same thing. Log sum, very similar, right? Because this is 0. It didn't really, this low feature didn't contribute very much. Linear model didn't do that well. On the other hand, when the problem occurs in high intensity, so this is another protein where the high intensity feature has outliers. So this is where the log of sum is really doing poorly, right? So when the true-false change is 1, comparing, again, condition 3 to condition 1, so log sum is doing very poorly. And this is because it will be really sensitive to outliers in high intensities. And so the proposed is a combination of Tukey median polish and censoring imputation, this two-stage thing, right, where you impute missing values using survival model and not just like using 0 or something else. And well, of course, when you have everything, so when you have both low and high, so this is comparing condition 3 and 1. So you see that it can go both ways, right? It depends a little bit, but essentially, in this case, well, of course, we picked the best examples, right, to illustrate that. But this is a case where imputing missing values and being robust to outliers, we are more on target than with other methods. So this would be the kind of spot checking a little bit the results, but I have more evaluations later for that. Okay, so this is in terms of the summarization, but we still haven't gotten to the point where I want to compare the conditions, right? So the model, if I just kind of go back real quick, so the model expresses just conditions, and if you have in this three-way factorial example with cancer invasive cell lines, well, we will have all these combinations of factors, so 9. This will be different conditions. But we just said that we wanted to answer complicated questions, such as, is the response to oxygen treatment the same for low invasive lines as for high invasive lines? So how can we do things like that? And so what we do internally, and this was really kind of helped us develop tools that are general, is to use each combination of conditions as here. So in this case, it will be 9 conditions, right? So each combination of treatments will be unique condition. But then if we want to compare, for example, high invasive normoxia at 6 hours to low invasive normoxia 6 hours, what we actually do, we extract the coefficients of the models. So this was kind of in the full model, but the new approach works similarly. So you extract the appropriate linear combinations of the appropriate parameters that help you answer this specific question. And the good news is that you don't need to do, as a user, you don't need to do any of that. So the user tells us, I want to compare this condition to this condition, or the average between these conditions to the average between these conditions. And the tool goes in the back and calculates all of this automatically. So we work out the coefficients of the linear combinations in all generality for that. And in some simple cases, when we have balanced data sets and all of that, you can actually derive specifically what the distribution is. In other cases, it's more difficult to say analytically, but you can always get to the actual test statistic and to the actual p-value for the comparisons of interest. And the last piece, of course, is the experimental design. And if I am to think about a question that people ask me most often is, how many replicates do I need? And so, and of course, my answer is it depends, right? And of course, it doesn't help people very much. And so what we do now as a kind of matter of, trying to be systematic about things, we view every data set that is being analyzed, automatically we view it as a pilot experiment for something else. So we say, well, what if our user will follow up on this experiment with a new experiment, which will look the same, in the sense that it will be the same technology, the same type of samples, but maybe they will have different conditions slightly, and so on. So we use this big model and the variance components of this big model And this is enough information for us to do sample size calculations, where we can now report for the desired fold change. This will be a number of biological replicates per group. And of course, the specific number will depend on the biological system, but at least it will give some guidelines to the user of what to do next.
So is this real?
It was one of the data sets, but you know, it really depends on the variance components, right?
So that means for 1.5 fold change, which is what about
Mhm.
you
Mhm.
recommend 20.
This was some yeast sample, you know, so it depends on your system, right? If you're doing cell lines, it's one thing. If you're doing yeast, it's another. If you're doing, you know, purified mixtures, it's something else. So I would not claim anything about these numbers specifically. I would say take your data, run it through the analysis, derive your own sample size calculation. I would very much stay away from taking somebody else's sample size calculation and thinking that it will apply directly to you. It's not at all the case, right? Because the variance components are very specific to the organism, but also to measurement technology and to everything else. So, well, so we work on the log scale, right? So we look at the difference between conditions. But if you it's actually a really good point. So you don't know. And well, plus, you know, I haven't said that, but we actually have many proteins, right? And different proteins have different fold changes, right? So this will be the smallest fold change that you can detect. So, well, there are two ways to do that. So one is if you have some literature which indicates that probably there will be some change for this protein, right? And then you say, okay, to detect, so if this is the smallest fold change that is of interest to me, based on the literature. So, you know, maybe 1.2 is too small for you for all practical purposes, right? So it's not necessarily what is the true fold change, but what is the minimal fold change that will be of interest to you to follow up. And then it gives you the sample size. But often people use it in the other way around. They say, okay, I have 50 samples. What would be the smallest fold change that I can detect for that? Let's say 1.3. And then you can ask yourself, is it worthwhile? Or maybe should I have more, spend more money to get lower, right? So you don't know. And you know, everything with sample size calculation, experimental planning, it's just a guideline, right? Because you are trying to essentially predict the future, right? Well, how good are we at predicting future? Who knows what happens, right? So you think your new experiment will have the same variability as your last experiment. Something changed and it's different technical variation, right? So everything is just hypothetical, right? But at least it's an educated guess.
So do you, by fold change, do you mean just ratio?
Ratio.
Okay, not log?
No, no, no. So, well, you know, because we're working with difference of log intensities, kind of summarized, and you just take the anti-log. So we usually, you can actually show this on the log scale, but most our users, they like to look at the ratio, and so that's what we do. Yeah. I think there's an option there, actually. Yeah. But to go back to that, so it is an educated guess, right? So you should still expect that things can go wrong, but at least it's not like just doing this and hoping for the best, right? So at least there are some kind of considerations for that. So that's, and maybe one thing I should like to point out is that something which is often kind of underappreciated a little bit is the fact that the number of proteins that you quantify actually has implications for the sample size. And you can show this from many perspectives, but one in our context here, because we talk about comparison between the groups and testing and p-values, so we run into the multiple testing problem, right? And so usually we need to adjust for multiple testing, and adjustments, if you have heard of Bonferroni corrections or Benjamini-Hogberg, so this type of corrections, they essentially require us to make stronger, to see stronger evidence against the null hypothesis to claim significance, just because we have more proteins. And so this is something, this is not part of the MSstats output, I just put it here just to make the point that if you have only one feature, you have this sample size, but if you have more features and 1%, let's say 50% of them changes, you need larger sample size. And if only 1% changes, you need even larger sample size. And the reason for that is that the sample size calculations are such that They try to protect yourself against false positives. So if everything changes, well, there's little opportunity to make false positives. So you don't need to work as hard, you need smaller sample size. If only 1% of protein changes, you really need to work harder to protect yourself against false positives. And this means larger sample size.
Well, what should we plan?
Sorry?
As a biologist, I'm not really interested in like even 1.5-fold changes. So this is very heartening thing because your single feature line tends towards 0. So if I'm looking for a 5-fold change, can I have just 2 replicates?
Well, you know, if you have a 15-fold change, you can see it by naked eye, right? Yeah. So at that point, it's actually a fair statement that the p-values are nothing else than a summarization between the variability and the size of the effect, right, between the conditions. And so if you are confident in your variability and if you're confident in your change, you don't need that many replicates. The problem is that your variability, it doesn't come from outside, it's estimated from the same data. And so with a small number of replicates, so unless you have some prior information of the variability, if you have to estimate this from 2 replicates, you tend to underestimate that. So you have this extra aspect of accuracy of estimation of variation in addition to just kind of technicality of how we go to the p-values. So, and then other things can happen, right, such as things fail, because again, this is theory, right? This is based on that everything will go according to the plan with these variance components. And real life is not like that, right? So this is kind of also a protection against other things which are not factored into that. So I would certainly do more than 2 replicates, even if you expect a very big fold change. Yeah. But it probably means that you don't need 100, so which is also good news. Yeah. Okay, and just to show you some extensions, so we just mentioned that like other experimental designs require other models. For example, time course would require some adjustments. So what we do in terms of the analysis is that we have some notation convention. So we ask the users to give unique names to each subject and to conditions. And then we can, from the notation, see if it's a time course design or not. And so we parse this, and we will fit the right model based on the design. And another extension is using labeled experiments. So if you spike in labeled references, so as I mentioned, we now have 2 peaks per run and 2 sets of fragments per run. And this is very useful. So maybe just very briefly, I can comment that if you have a lot of run-to-run variation, you want to compare the endogenous blue peaks to the endogenous red peaks. But run-to-run variation inflates this so much that you cannot really make any statements between the difference between the groups. But if you add now reference peptides to each of the endogenous peptides, you see that this variation is really technical variation, because reference peptides have no biology in them. And so by looking at the deviation of blue dots from the black dots, you see that in the disease sample, they're systematically larger than in the healthy sample. And this allows you to see that this difference is more systematic than as expected by random chance. So labeled references allow you to reduce some of the technical variation. In statistical language, it's called blocking. And the kind of aspect of this for us is that it now makes the data structure more complicated, because now we have the reference counterpart for every endogenous counterpart. And we can extend it the same way. So now we have reference intensities in addition to the endogenous intensities. And again, we extend the model, and we also analyze the subplot part separately. We have a summary of the protein abundance in each run, which eliminates this run-to-run variation. And then we proceed working with the biology itself. So it's an extension of that. So I would like to tell you a little bit about the tool itself. So it is interfaced with Skyline, which is an external tool for a more general kind of computational workflow for processing the signals from the spectra. It's available in Bioconductor, so it's not even up to date. So we have a pretty wide user base at this point, which makes us very proud, but it also a lot of responsibilities. So we work very hard to make sure that things are working as they should. We do a lot of kind of educational efforts, and we offer short courses, usually in combination with Skyline teams, so that we can train people in both signal processing and understanding the raw data, and also the downstream statistical modeling. So one of the courses that will take place at Northeastern will be the second time that we do it. I'm very happy about that. So now it depends how much time I have, because I have a few more results. So David, what can I do?
Yeah, maybe another 5 minutes.
5 minutes. So, well, let me, you know what, let me just take a break actually and see if anybody has questions. Maybe that would be a better use of time. TMT and SILAC is very high on our priority list. So So SILAC is a very straightforward extension. TMT and iTrack will be a little bit different because MSMS spectra have the stochastic grouping of the reporter ions, and we will have to modify the methods more. But SILAC, you know, so it was just a combination of waiting for an opportunity for this and for funding, but it is very, very close to be there. Yeah, we definitely know that it is important, yeah. It would be very easy to do. We haven't done it yet. So if you are familiar with R, you can just, so it's open source, right? You just take the code and just add a confounder there, right? Yeah, this is something we've been thinking about. So the problem is that it would require a different input format and a lot of input checking, so we haven't gotten there yet. But technically speaking, there is no reason not to do it. It's very easy to include. Any? Well, actually, yes. Well, yes and no. So actually, yes. So I didn't show this. I think I had this is just an example of volcano plot, right? So this is where you have a comparison of two groups, right? But what often happens, you compare different pairwise comparisons of different types of conditions. And so what you do then is we provide a heat map of p-values and significance, right? And so you can cluster proteins by their pattern of change, for example, up in all comparisons, up in a subset of comparisons, you know, going down and so on. So that is automated there. If it is not what you look for, so another functionality that we have, I didn't talk about, is what we call run quantification or condition quantification. So what we do here is look at the differences between conditions using linear combinations of parameters. But you can do the same thing to quantify just one condition and not just comparison of conditions. And so what we also do, we can just have one number, like in the subplot, we have one number per protein per run, but we can also use this linear combinations of parameters to have one number per condition per run without comparing it, right? And so if you want to draw heat maps with some clusters, you know, just to see how the proteins behave in individual conditions, so that would be the output where you just have a summary on some relative scale of the protein abundance in one condition, or in the individual replicate, whatever you need, right? And then you can just use your own clustering or something like that. Okay. So let me then maybe just take 1 minute and show you some additional benchmarking. And I think, and this is a bit of a reality check for us. So as I say, our wish is to be general and be useful in a variety of circumstances. And so one way to accomplish this is to benchmark this across a large number of data sets and make sure that we do not make mistakes across the board. And so we spent a lot of time actually doing this in the last year. And so we, by having many collaborations with experimentalists, we have accumulated a fairly large collection of controlled mixtures, such as the IPRG mixture, where you spike proteins in known concentrations. And so we now start using this to compare different tools and different results. And of course, we also want it to be independent of the signal processing tools. And so maybe I can start here. This is the IPRG data set that I just mentioned. and it was originally processed with Skyline. And so it's a bit complicated picture, so I'll try to just walk you through that. Different columns are different statistical methods based on the signal processing and Skyline. The rows are different true fold changes of the proteins, and we know they're true because we spiked proteins in this way. Now, in green is the sensitivity. And what we did, because we didn't really want to directly compare tools of fold changes, and also higher fold changes are obviously easier to detect than others, each row is normalized. So dark green is the most sensitive of the 4 methods for this fold change, and then lighter green will be how much less sensitive it is. And so here the leftmost column is the proposed method. The second one is robust summarization without imputation of missing values. Then this is our older linear model, and then log sum. This is what kind of many kind of existing methods also use. And so essentially darker is better. So this means more sensitivity. And then here this was one fold change that nobody could get, so we just kind of highlighted it like in this. And then the blue is the specificity, and in this case we used the background proteins, and it was specific. So here you have the number of comparisons, so many background proteins here, right? And then this is the proportion of missing, so you can kind of, each fold change in each data set is characterized by the proportion of missing data. And then, as I said, we want to make sure that this is not dependent on the tool, so we process the same data with MaxQuant and then also with Progenesis. And so the reality check is it's really hard to say that one statistical analysis tool is much better than another across the board. So there are always some configurations of some types of variation or some decisions by signal processing tools which go towards one tool versus the other. But we felt like in this example, more often kind of, well, it was doing the right thing. And it's also kind of an interesting aspect also to see how different tools have different proportion of missing values. So it definitely affects their decisions.
So MaxQuant seems to be doing pretty well.
MaxQuant is doing quite well, yeah. But again, you know, just to kind of clarify, right, that it's always normalized within the row, so it's not an absolute sensitivity. And they were very careful, because otherwise, fold change of 32 will dominate everything, and then everything else will be much worse. So it is only within each row relative to the best method in the row, right?
So also not across the section.
Exactly, and because really, at least for the time being, we are not interested in comparing the tools, we're interested in comparing summarization methods and statistical methods. However, it was actually quite interesting, because when we did all of these method comparisons, and so this is a very old data set that we had long time ago, which was processed by different tools, and this is from the Jürgen Cox paper, LFQ paper, more recently. So what happened, so we haven't had much kind of conversation with MaxQuant, but with Skyline and Progenesis. So when the authors of the tools see these results, it actually motivates them to also find out what's going on. And we actually spent a lot of time going back to signal processing and trying to understand. And this figure actually survived several iterations, because the tools actually decided to also make changes based on our feedback. So this was quite nice. So this is the sensitivity, and then the other is the estimation, because, you know, sensitivity is testing, right? Significant or not. Estimation is how close the estimated fold change is to the true fold change. And we do the same thing here, so it's also normalized within the row, right? So it will only be how much, so this is the mean squared error, is the deviation from the true fold change and the estimated fold change squared on average over all proteins which happen to have this fold change. And again, darker is better, but only within rows, so we don't compare across things. And again, we feel like not always, right? Sometimes there are some configurations where who knows what happens, but generally speaking, this approach also works well in terms of estimation. And we did the same thing for DIA. So for DIA, it's also kind of very similar type of approach. And clearly you see that lower fold changes are more difficult, higher fold changes are easier for everybody. And then we had the same thing for SRM. For SRM, testing was kind of easy for everybody. There was really nothing to say. Estimation, there's a little bit improved the accuracy of estimation as compared to that. So I will have to stop here. So I had I wanted to show you a case study of how we actually used MSstats for a clinical study. And the interesting thing was that it was a fairly large number of patients, so it was interesting see, but maybe next time. Maybe just the very last sentence is that we actually have a lot of extensions to MSstats in the pipeline, which are very close to being kind of used. And if you go to US HUPO next week, we will present some of this, like in posters or in some kind of other forms. And so there's 3 types of extensions that are also based on the feedback by the users, by the way. One is statistical process control, which is designed for large-scale experiments where you profile large number of samples. And you want to be able to automatically diagnose deviations from the performance according to specifications. And so how do you essentially see some deviations? In this case, this is based on the peak area, but there are many other readouts. And so how do you ring the alarm bell soon, but not too soon, right? And so how do you do that? Assay characterization in terms of limit of detection and limits of quantification. We have some interesting implementations there, and also relative quantification of post-translational modifications. This was also one thing on the wish list of many of our users, so we're also making progress towards extending MSstats to that. And so I'd just like to thank my group. So a lot of people actually contributed to the development, but maybe I should single Mina Choi, who is the lead developer of MSstats, and Mike Cheng, who is a co-developer, and now Tsung Heng is the second in charge for MSstats. Eralp leads the process control project, Cyril leads the limit of detection project, and Tsung Heng also leads the protein characterization project. And I also have to thank the collaborators, especially from Mike McCaw's lab, Brendan McLean, who is the author of Skyline. It has been a really very, very exciting collaboration, very beneficial to us, and I hope it's mutual. And also the Abersold lab, who really gave us a tremendous amount of feedback and data and suggestions on how to make this better. Thank you very much.
Thank you for coming. Maybe we can take 1 or 2 last questions, if anyone has.
So the spiking samples themselves have also some error.
Of course.
Okay, so that doesn't mess you up.
We assume that the stated fold change is a true fold change, because, you know, that's kind of our starting point, but clearly this can have errors.
Okay, great. Let's thank Olga again.